Students, in the previous video we have seen the application of biotechnology in medical field. Prior to that we have seen application of biotechnology in agriculture to produce insect resistant plants as well as pest resistant plants. So we have studied about BT cotton as well as we have studied about parasite nematode resistant plant. Then we have seen the application of biotechnology in medical field. So in medical field it is used, the techniques of engineering is used either to treat a disease, diagnose a disease or to prevent the disease. So for treatment purpose we need therapeutics. So we have seen the application of biotechnology in production of therapeutics, and one of the example that we have seen is genetically engineered insulin, right? And we have seen the technique of genetic engineering for curing the disease. So the technique itself is used for curing the disease, that is gene therapy is used, wherein the non-functional gene is or the deleted gene is removed and we can insert a functional gene using genetic engineering technique in order to once again fulfill the normal functioning of those genes, right? So we have another application of biotechnology, that is in molecular diagnosis. That is molecular diagnosis. The techniques of biotechnology are used to diagnose various diseases. It is used to diagnose various diseases. Why we want diagnosis? Before the treatment of any disease, it is important to diagnose the causative agent or reason for that disease. Yes or no, students? If you know what is the reason for the disease, then only you can approach to the efficient procedure for the treatment purpose. For that purpose, first is we have to diagnose a disease or what is the causative agent for this disease. So therefore the biotechnology is helping us or allowing us to use the molecular techniques in order to diagnose the particular disease. So early diagnosis and study of pathophysiology is very important for effective treatment of a disease. That is, if disease is not diagnosed at the earlier stage, then it will lead to the advanced stage. At advanced stage, it is difficult to control the disease condition. So if you are able to detect a disease in the early stage itself, then the diagnosis is done earlier stage, the treatment can be initiated at a very early stage, and when the infection is at very little rate, then only the treatment can be initiated, which can destroy the infection completely and it will prevent it from occurring in the future. But if the early diagnosis is not done, then the disease condition will be at advanced stage. That time when we are approaching towards the treatment process, maybe the chances of curing would be very less. For that particular reason, the molecular techniques help us for early diagnosis and also help us to study the pathophysiology. What is this pathophysiology? Pathophysiology means there is abnormality in the biological functioning of the body. See, a normal functioning is known as physiology. There is a procedure in the metabolic pathways. After this step, catalytic activity, another catalytic activity mediated by a particular enzyme must be carried out, right? In case if there is any invasion by a foreign body in the human body, any antigen is being invaded in the human body or any foreign body enters into the human body such as viruses or bacteria, the physiology is going to change. The normal biological process will be disturbed because of the infection, and the bacteria or virus also draws its nutrients from our gene machinery. So that is the reason what will happen. There is a abnormal biological functioning taking place in our body, which is referred as pathophysiology. Change in the physiology because of the invasions of a pathogen, that is known as pathophysiology. So if you are able to study the pathophysiology at the earlier state only, or if you are able to find out the causative agent for this particular disease at the earlier stage only, then the treatment can be more effective. So the DNA of these pathogens can be detected in early stages when no much symptoms are developed, and helps to provide best treatment for high efficiency of curing, right? Because the organisms act at the molecular level. Yes or no? Why we need require molecular techniques or biotechnology techniques to diagnose a disease? We in the ancient days we have serum analysis and urine analysis, where they collect the urine and they perform certain tests, and then they give you a report after 1 day or 2 days. Okay, so what will be there? That is going to increase your risk of disease. It is going to increase the risk of disease because of the delay in testing process. If we are able to detect these pathogens at early stage, then we are going to have an efficient treatment for the disease. See, if you are using the urine analysis as well as serum analysis, that is not going to provide early detection for most of the high risk diseases such as HIV, AIDS, cancer. Okay, you cannot detect those diseases through urine analysis and all. So what we have to do here, the molecular techniques plays a very important role. Okay, that is going to detect these infections at early stage, right? Because the organisms act at the molecular level. Now for any disease to provide a symptom, if any organism, bacteria, virus has invaded the human body, so before it could provide the symptoms, what will happen? The bacteria has already entered the body. Now for coronavirus, they are giving the window period of 14 days, incubation period. Why? The symptoms can develop anytime in these 14 days. Right, students? So, we have to be, now why this rapid kit is there for coronavirus, which is going to detect at very initial stage only. So, it is going to analyze the antigen present in it, invaded in it. Okay, or else you can detect the change in the DNA. So the symptoms will produce whenever the foreign body invades the human body and human cells, and is going to change the DNA. A new combination of DNA will be formed. Okay, it is going to attach its DNA to the genomic DNA, and therefore a new combination of DNA will be formed. This new combination of DNA will code for certain proteins, and those proteins are going to be produced in the body, and that is going to produce the symptoms or allergic reactions. When you get those symptoms, then only you go for a treatment, right? But in case of suspected patients, if we think that we have interacted with a HIV patient or with a COVID-19 patient, then you are known as a suspected patients. Means you are interacted, so there is a chances of getting this type of infection. So what we do for that purpose? We go and give it a test. We are going to give for a test our samples, which is going to check. So here in the genetic engineering technique, the DNA will be taken and it will be tested before the symptoms are produced, before the infection is spread. Okay, early diagnosis is possible with the molecular techniques. So some of the techniques such as PCR reaction, ELISA, that is enzyme linked immunosorbent assay, DNA sequencing are some of the early diagnosis methods, as well as DNA probes are used to detect defective genes in order to proceed with the gene therapy. Okay, and autoradiography techniques are used, and whatever the techniques you have studied in biotechnology principles and processes, agarose gel electrophoresis, PCR reaction, these are all used, okay, for early diagnosis of a disease. PCR helps to detect the low concentration of pathogen. See, when initially a pathogen enters into the body, before the infection could spread, there will be small amount of pathogen, a little concentration will be present in your blood. That can be detected by PCR technique, because PCR, that is polymerase chain reaction, is a amplification technique for genetic engineering. You can multiply the gene of interest to billion times or million times through PCR reaction. So the PCR reaction helps to detect the low concentrations of the pathogens such as bacteria, viruses when there are no much developed symptoms are present. Right? And apart from that, we have another technique known as ELISA, that is enzyme linked immunosorbent assay. It is enzyme linked immunosorbent assay. Here we are going to either detect the antigen or antibody. Whenever a pathogen is invaded, okay, what will happen? The body is going to produce antibodies in response to the attack by these pathogens or antigens, right? So either you can collect the antigens from the blood sample or you can collect the antibodies, and then we can detect how much the infection is present and what type of infection is present in the body. That is done by ELISA technique, that is enzyme linked immunosorbent assay. Now the PCR is having different types of applications, okay, in the molecular diagnosis. One is it is used to detect low concentration of pathogens in the body, okay, such as HIV suspected pathogens. When suspected, you still don't know whether it is positive or negative. Clear? So if you are already contacted with a HIV positive person, then there could be chances of transmission of this HIV. So they are known as HIV suspected. So in that we can check whether there is a infected genetic material is present or no through PCR technique. And PCR detects mutated genes causing cancer in suspected patients. Okay, in some patients, if you are thinking that there could be some mutations, and there also you can detect the mutated genes using PCR technique, that is polymerase chain reaction. And PCR used to detect genetic disorders and also in paternity test. Okay, that also used for in forensics for paternity test also PCR reaction is used. Now let us see how this PCR reaction is used to detect the concentration of pathogen in the HIV suspected patients, or how this PCR is used to detect the mutated gene in cancer suspected patients. Okay, let us see step by step. PCR is used for testing the suspected AIDS patients, right? Now how we know that one of the important purpose of this PCR is amplification of the DNA. It is going to amplify the DNA. What will happen in a person who is affected with the HIV virus? How the disease mechanism is going to take place in that particular person? See, whenever the HIV virus invades a person, okay, it is going to convert the genetic material of the person. It is going to convert the genetic material of the person. So, the infection is with the RNA virus, and therefore it converts the DNA of the human body with the infected DNA by reverse transcription. What happens? The virus is having RNA as the genetic material. So through reverse transcription, it is going to produce the enzyme reverse transcriptase and integrase enzyme, which converts the RNA into DNA. First, the virus genome is getting converted to DNA, and this DNA gets integrated with the host genomic DNA. The affected cells are host genome. So it is getting integrated with the host genomic DNA, and new combination of DNA is formed. And a new combination, which is known as a recombinant DNA, is formed. Now we can use PCR reaction because there is a new combination. The pathogen has entered. So we are going to use to detect the nucleic acid of the host cell. Yes or no? PCR reaction will amplify the nucleic acid of the host cell. So whatever the new combination of the DNA formed in the human cells can be detected using the PCR reaction. So what happens in PCR reaction? There is a step-by-step reaction. First is denaturation process, and second it is annealing process, and third it is primer extension or chain elongation process. So in the first step, what will happen? Denaturation. Whatever the sample we have collected, that is recombinant DNA or new combination of DNA, is used as a source DNA in the PCR reaction, and then it is denatured at very high temperature at 96 degrees centigrade. After that, what will happen? Next is annealing. In annealing, we are synthesizing the primers. So we already know in the research and development, by research we already know that for the HIV virus, what is the complementary DNA? Yes or no? Through reverse transcription in vitro, we are going to produce from RNA to DNA is produced, and for that DNA, what is the complementary DNA is also formed. So we are using certain primers. Primers are nothing but they are the short segments of DNA or the oligonucleotides which are chemically synthesized, contain 20 to 30 base pairs. They contain 20 to 30 base pairs. Okay, so we are designing complementary primers that are specific to the this infected DNA or the HIV DNA. Getting students? HIV virus has RNA as a genetic material. So through reverse transcription in vitro, we are producing the DNA, and for that DNA, complementary DNA is prepared, and that complementary DNA is used as primers. Okay, and we are allowing in the next process in the PCR reaction is annealing. So the temperature is lowered to 45 degrees centigrade, and then these specific complementary primers to the HIV virus are allowed to bind. They are allowed to bind to the denatured DNA. Right? Okay, if the patient is having the positive, that is HIV, it is infected with the HIV virus, then these primers will go and bind to the complementary regions in the infected DNA. Right? Okay, and then the next step will be primer extension. So the Taq polymerase enzyme will add the DNTPs and extend the chain, and it will amplify the DNA. Later you can see how much amount of this DNA is present. Okay, how much? One is when the primers get annealed, then it is understood that the person is positive, right? Because they are specific primers to HIV virus only. If the person is negative, then those primers will not find the complementary region there, so they do not bind. So how you will get to know whether it is a positive or negative after the reaction? We are going to perform the agarose gel electrophoresis. Agarose gel electrophoresis is performed to see the amplification of the DNA, whether the initial source DNA what we have added, and then after the reaction what is the DNA we have got. If it is amplified, then it will show a thick band, then it is going to show the large molecular size when compared with the source DNA. That is how we are going to detect whether it is a HIV positive or negative using the PCR reaction. And you can also using this PCR reaction, you can also say how much amount of infection is present, whether it is every time stage 1, stage 2, different stages are there for this HIV infection. So every time when you go, they perform the PCR reaction and then they check for the amount of infection by PCR reaction, the size of the DNA, whether it is increasing or it is decreasing after the PCR reaction. If it is increasing, then the infection is at very high rate. If it is decreasing, after the different therapies will be given like chemotherapy and all. If it is decreasing, then the molecular size is also going to be reduced. This can be found in the agarose gel electrophoresis. So this is how PCR is used to detect the HIV in suspected AIDS patients. Another application of PCR reaction in molecular diagnosis is it is used to detect mutated genes in cancer. It is used to detect mutated genes in cancer. Sometimes cancer is the uncontrolled cell growth, Uncontrolled proliferation of the cells is going to take place that results in the tumors. Sometimes due to the genetic disorders, that is due to the mutation in the genetic material also, the abnormal cell division can takes place resulting in the cancer. So we can detect those mutated genes also through PCR reaction. Not only the PCR reaction is going to detect the mutated gene. After PCR reaction, there are certain steps that is going to detect the mutated gene. The first step is in PCR reaction, we are going to take the from the blood plasma cells, we are going to take out the DNA, the genetic material we are going to take out, and then this is purified. Isolation of genetic material is done from these cells, and whatever the DNA we are getting, that is used in the PCR reaction. So after the isolation of the genetic material from blood cells, it is fragmented using restriction endonucleases. Fragmentation is going to takes place, and then specific primers are used for the amplification purpose, and they are amplifying the DNA. Once the DNA is amplified, okay, next is the probes are used. Hybridization of the probes is going to takes place. What is this hybridization of the probes? Probes are nothing but they are single stranded DNA or RNA. Single stranded DNA or RNA that are complementary to the normal DNA. Now we don't know where is the mutation present in order to design the specific complementary DNA. So we are going to have we know in a human body what is a normal DNA sequence. So accordingly the complementary DNA strands are produced. Okay, these single stranded DNA or RNA which are tagged with the radioactive elements, which are linked with the radioactive elements, they are known as probes. They are known as probes. So these radioactive elements in the reaction, during the process of reaction will emit the light. So based upon that corresponding light, we are going to detect whether there is a mutation or no. Or this can be observed in a particular film known as X-ray film. There also we are going to, if they are appearing, okay, then they contain the radioactive elements. Right? For that particular purpose, the probes are used for the detection purpose. Okay, the probes are tagged with the radioactive molecules and they are complementary to the normal cells. Okay, now whatever the PCR amplified product is there, okay, the probes are allowed to hybridize with the complementary DNA in the clone of cells. Okay, the PCR product contains the amplified DNA, okay, which is then restriction endonuclease will act upon it, okay, it will fragment it, and now the probes are allowed to hybridize. So the probes will seek their complementary DNA because we have designed the probes or the single stranded DNA that is complementary to the normal DNA. So in the whole genome, certain part will be mutated, but the rest of the part will be the normal genetic material only. So these probes will bind to their complementary regions. Okay, they are going to hybridize, that is known as hybridization. They get hybridized with the complementary DNA and form a hybrid DNA will be formed. Next step in this is we are going to detect these probes. How we are going to detect? By a process known as autoradiography. By a process known as autoradiography. What is this autoradiography? That is, we are detecting these probes in the X-ray film. Okay, in the X-ray film, or it is known as photographic film, which is going to generate the image. Which is going to generate the image that is used for detecting these hybridized probes. Right. So after the pairing, these are allowed or they are immersed in a matrix. These radioactive molecules along with the DNA samples together it is going to be immersed in a matrix and then they are exposed to the X-ray film or photographic film. Okay, since these are radioactive elements are present in this, so when they are exposed to this X-ray film or photographic film, it is going to form the image. It is going to generate the image. Okay, and then we can find out which gene is mutated and which is not mutated. Okay, and they are already labeled. So you should know the labeling of these genes in the prior only. Before you could introduce into the matrix, the samples labeling with radioactive molecules must be unique, that it should be different and they should be known before to you. Okay, so after the detection what will happen? Whichever the probes containing the radioactive elements and find its complementary DNA in the clone of cells is going to appear or form the image in the X-ray film. It is going to form the image in the X-ray film, and the others one, okay, for the mutated gene, it will not find the complementary probe because we have designed the probes in accordance with the normal DNA, and for that particular purpose the mutated gene will not find its complementary probe, and therefore that will not be detected in the X-ray film or the photographic film. That particular DNA sequence, okay, it will be studied through DNA sequencing method, and the mutation can be which gene is responsible for mutation will be detected. So this is how the PCR reaction along with the autoradiography will help to detect the mutated genes causing cancer. Right? And last we see one more molecular biology technique, that is ELISA. ELISA is enzyme linked immunosorbent assay. It is enzyme linked immunosorbent assay. ELISA test is used for detecting HIV also, as well as it is used to detect other pathogen causing diseases also, wherein the antigens and antibodies are produced in the body. So, ELISA test, that is enzyme linked immunosorbent assay, is a quantifying method. You can quality testing also is, quality means presence or absence. Quantity means how much is present, can also be detected through ELISA test. So, it is used to detect antigen of HIV virus, okay, in the HIV positive patients. So, whenever there is an invasion of the HIV virus, it will produce P24 protein which act as antigen. It will produce P24 protein which act as antigen. So, once the antigen is produced in the body, accordingly in response to it, the specific antibodies are also produced in the body. So, what will happen? You can either detect the antigen or you can detect the antibody through this ELISA test. So, the reactants could be antigen or it could be antibody. What is the principle behind this ELISA test? What is the principle behind this ELISA test? It is working based on antigen-antibody interaction. It is working based on antigen and antibody interaction. So, you must all be aware that a specific antigen will interact or makes a complex with a specific antibody only. Right? A specific antigen will bind to specific antibody only, like how the enzyme substrate complex will form. A specific enzyme will react with a specific substrate only. So, even the antigens also are specific. Antigen antibody interaction is also specific. So, in the ELISA test, if you want to analyze the antigens or if you want to analyze the antibodies, okay, after the formation of the antigen, the antibodies are produced. So, in order to find the amount of the infection in the body, we can isolate these antibodies from the blood cells and then we are going to allow it to interact with the specific antigens. Now, we know if the person is with HIV positive. So in the quantification, how much infection is present, to know that these antibodies are immobilized on the solid surface or microplate. Okay, a microplate is used in a molecular biology labs and all. So here we are going to immobilize the antibodies. We are going to immobilize the antibodies, and then it is allowed to interact with the counterparts. So if you are immobilizing the antibody, then we are allowed these antibodies to react with the antigens. Or if we are immobilizing the antigen, then they are allowed to react with the antibodies. Right? So we are using the antibodies. Now you are suspecting a particular disease will be present. So that particular pathogen, whatever the antigens it is causing, those antigens are allowed to react with these antibodies. It is allowed to react with these antibodies. Right? Now, whatever the antigen you are allowing it to react with the antibodies, they are in turn linked with the enzyme. So this is enzyme linked immunosorbent assay. Right? We are immobilizing the antibodies, that we are going to quantify the amount of infection present. So we are immobilizing the antibodies, and we are allowing these antibodies to react with the specific antigen. Right? So you don't know particularly which antigen is present in case of qualitative analysis. In case of quantitative analysis, you know which disease is present. So accordingly you are going to use that particular antigens. Right? So the antigens are used. Okay, let us say in case of the suspected patients, we don't know. So we use different types of antigens. Okay, we are allowing them, but in turn these antigens are linked with a particular enzyme. Why? Because enzyme will react with the substrate to form the product. Enzyme will react with the substrate to form the product. Okay, and when these antigens are allowed to bind to these antibodies, after certain period of time of interaction, this particular reaction mixture is washed away. It is washed. Why? Now only the specific antigen and antibody complexes will be present, and unbound antigens or unbound antibodies will be washed off. Because if they also contain, because we have used different antigens with which are linked to enzyme. Though they are not bound to the antibody, when they are allowed to react with the substrate later, they will produce the color or product. We are going to quantify that product only. Based on the quantification of the product, we are going to tell the intensity of the infection. Right, students? So, after this washing process, then you will have a specific antigen antibody complexes only, and unbound antigens or unbound antibodies are removed through washing process. Now next, this particular product, antigen antibody complex, wherein the antigen is linked with the enzyme, they are allowed to react with a particular substrate, specific substrate. So the enzyme will be having a specific substrate. When they react with a specific substrate, it is going to produce a color change will be produced. So we are going to quantify this amount of color change through a calorimeter, which is going to find out the amount of color, or you can use the UV spectrophotometer, wherein you can find out the absorbance, how much amount of color is present. The intensity of the color will be directly proportional to the concentration of the infection. Means how much amount of the infection is present is directly related to the intensity of the color change. More color means more infection, less color means less infection. Clear, students? So in some cases the antigen is linked with the enzyme, but in some cases another antibody is used known as secondary antibody. So these secondary antibodies and these enzymes, they are known as reporter enzyme. It is known as reporter enzyme, which is going to give you the conclusion of the presence or absence of the infection. Right, students? Once again I will tell, in ELISA technique, it is based on the principle antigen antibody complex formation. Okay, so if the antigen and our antibody, the specific antigen antibody will bind to one another, and if your externally passing the antigens, then antigens are linked to the enzyme. If we are using antibodies, then antibodies will be linked to the enzyme. That is one way of detecting. Another way is, after the antigen antibody complex is formed, they are allowed to bind to another specific antibody. This specific antibody is known as secondary antibody, and this secondary antibody will not bind to the antigen, but it will bind to the antibody only, wherein you already know what antibody it is. Right, students? So this is used to find out the intensity of the infection. So these are the three techniques, that is PCR reaction is used, and ELISA technique, as well as a DNA sequencing and autoradiography. These are some of the molecular biology techniques that are used for diagnosing the disease at early stages. Right? As well as you can, among all this PCR reaction, ELISA are the major important molecular biology techniques which detects the disease at early stages.